iridescent light waves demonstrate constructive interference, then we can see the colors of those light waves. The colors change if the film gets thinner. In the case of bubbles, this is when they start to evaporate and pop. As the film gets thinner, the waves with longer wavelengths, like red, orange, or yellow, so all wavelengths become cancelled out. Chemiluminescence. We typically hear about bioluminescence, also known as the phenomenon explaining why fireflies glow, or why there's certain types of algae that light up in caves. From the name itself, bio, which means life, and luminescence refers to the emission of light. Put them together, and it's light production in living things. Now, instead of bio, we're going to tackle chemiluminescence, the more general form of bioluminescence. The easiest way to describe it is that it's a light emission phenomenon due to a chemical reaction. The more interesting parts are the how and why does it happen. First, how is chemiluminescence different from all other luminescences? Other luminescences like fluorescence or phosphorescence, the presence of light comes from an external source of light energy. The best example of chemiluminescence demonstrated are glow sticks. Pick up a glow stick, bend it a little, hear a little crackle, and it lights up for a certain period of time. Inside those glow sticks are two chemicals. One is contained by the plastic, rubbery exterior container, and inside the rubbery container is a small glass container containing the second chemical. That's why we have to bend the glow stick and hear it crack so that the glass breaks and the two chemicals mix. Now, let's consider. Chemicals 1 and 2 will react to form products 3 and 4. The atoms, molecules, or ions of chemicals 1 and 2, the reactants, will have to collide with each other in order to form the products. But of course, it's not that simple. The success of the reaction will depend on several factors, like increase in temperature, stronger concentration of the reactants, or introduction of a catalyst. In the midst of the conversion of the reactants into the final products, an intermediate state called activated complex forms when the activation energy is sufficient. Activation energy is the difference between energy between the activation complex and the energy of the reactants. If the activation energy is too high, then the complex will be volatile and temporary. On the other hand, if the energy is too low, it may not be enough for a successful chemical reaction. Within the activated complex, there is what's called a transition state, where the potential energy of the intermediate molecules is at its highest. The transition state is the indicator that the reactants are more likely to be converted into the products rather than the reactants being formed back again. In short, the transition state tells us that we're in for a successful chemical reaction. What we've been talking about so far is the process of chemical reactions. Chemiluminescence undergoes the same, except for a few details. What happens to some chemical reactions is that heat energy is released. But in chemiluminescence, we need light energy. Now, the product's vibrational state is excited. The heat energy spreads through the product, making it warmer. It's a similar process for chemiluminescence, except it's the electrons that become excited. The excited state is the transition state. When excited electrons calm down or return to the ground state, the energy is dissipated as a photon or a light particle. But we have to admit, we rarely care about this process. What matters more is that how do they produce different colors? Or can all chemical compounds produce this light thing? In the case of glow sticks, a dye compound is mixed in the plastic tube. So when the inner glass tube breaks, the color of the dye will start to glow. Different compounds producing different color pigments is another story. But simply, that's where the colors come from. Sometimes, the colors depend on the reactant compounds. Like any other phenomenon, chemiluminescence is only possible with a few special compounds, which have the properties to produce light. One of which is luminol, the one used in crime scenes to trace blood, because this compound reacts with the iron in the blood. Luminol, when mixed with an oxidant, usually hydrogen peroxide, produces this blue glowing light. For glow sticks, hydrogen peroxide gets mixed with diphenyl oxalate, or simply sialume, to produce this luminescent tube of light. Luminol and diphenyl oxalate are two of the few compounds that are capable of bringing out the amazing thing we call chemiluminescence. It's amazing to witness that light can be found in many different sources. Bioluminescence. If we hear the word bioluminescence, we think about fireflies or glowing moss. But we're going to have to dig deeper than that. If we're not familiar with chemiluminescence, it's basically a chemical reaction that emits light as a result. Bioluminescence is a form of chemiluminescence, only that it occurs in living things, based on the prefix bio, meaning life. Of course, not all living things are capable of glowing internally. Most of these special bioluminescent creatures are found underwater, and they use their lights for different purposes, like for signaling or mating. The light generated in bioluminescence is what we call cold light. Usually, when light energy is released, a huge amount of heat is also generated. That's why light bulbs are warm when they've been turned on for a long time. But in this case, cold light is when only less than 20% of the source's light energy generates heat. Being a cold light is actually important because higher thermal energies can affect other systems of the organism's body. Because bioluminescence happens inside a living thing, the chemicals or other reactants must be biocompatible. The generic term for chemical compounds that enable bioluminescence is called luciferin. Luciferin is the substrate, or the substance acted upon by a catalyst or enzyme in a chemical reaction. When the luciferin undergoes a catalyzed oxidization, it'll produce oxyluciferin. When oxyluciferin decays, it will release photons, and there we have our bioluminescent light. This enzyme that will catalyze luciferin is generically called luciferase. The luciferin and luciferase are different for each kind of organism. There's a certain compound and catalyst for the bioluminescence of a firefly, bioluminescent bacteria, ostracods, and other bioluminescent species. A relatively new discovery, however, surfaced that some reactions do not have a luciferase, or the catalyst that speeds up bioluminescence. Instead, luciferin reacts with what's called a photoprotein. But unlike luciferase, the luciferin and photoprotein also have to involve calcium, aside from oxygen, to produce light. Luciferin photoprotein reactions usually produce short-lived luminescence and usually need to recharge, unlike those involving luciferin-luciferase reactions. One organism that uses photoprotein is the parchment worm. Scientists are still studying other properties of photoprotein. Depending on the organism, luciferin can be self-manufactured or obtained through the food they eat via symbiotic relationships with other organisms. An example of an organism that can produce luciferin on its own are dinoflagellates, a kind of plankton-sized algae. They give off this blue-green light that sometimes makes bodies of water glow. Glowing dinoflagellates is a good indicator of the health and life of the bodies of water they thrive in. Fireflies already exhibit bioluminescent capabilities in their larvae stage, which is why they're called glowworms. On the other hand, some species of squid and fish rely on the bacteria they host on their organs to produce bioluminescence, demonstrating a commensalistic symbiosis. Most bioluminescent marine organisms emit blue-green light colors, because those are the most visible colors in deep oceans or seas. Plus, a lot of marine species also cannot process the other colors in the visible light spectrum. Insects, such as fireflies and beetles on the other hand, produce certain light colors, depending on the amino acids present in the luciferase. When the oxyluciferin decays, the energy from that decay determines the hue or color, and what determines the energy are the amino acids in the luciferase. Like any other special characteristics of organisms, bioluminescence is not just fancy lighting for animals and plants. Organisms' luminescence serve necessary functions for their survival, such as attracting and trapping prey, like what anglerfishes do. It can also be used for tricking or escaping predators, such as for squid species that are incapable of producing ink. Bioluminescence is also a form of aposematism. Bioluminescent creatures also use light to attract possible mates, just like what female fireflies do. And, as briefly mentioned earlier, bioluminescence can also help indicate the condition of the environment the organisms are living in. Gemstone coloration. Do you ever think about how different gemstones get their color? They don't have dyes or natural pigments because they're rocks. Light, of course, is a part of the explanation, but they really don't lose color. It's just that we can't see the color when changes in lighting occur. Several varieties of gemstones actually have the same composition and structure, but we are able to distinguish them based on their color. Now, we already know how we perceive colors through light, and this is going to come in again as we figure out how similarly composed gemstones get different colors. But to recap a bit, we remember that light is an electromagnetic vibration at certain wavelengths. The human eye can only perceive wavelengths that we know as the visible light spectrum. Now, if all the different wavelengths of visible light pass through a gemstone, it'll appear colorless. On the other hand, if the gemstone absorbs all light, it will look black. To get a specific color, say red, all wavelengths will be absorbed except for the red light wave. Then the gem will appear red. There are reasons why the various gemstone varieties absorb light differently. First is because of the coloring agent, and there are two types: idiochromatic and holochromatic. Idiochromatic gemstones are those referring to those that deliver their color from elements that are necessary for their chemical composition. To put it simply, idiochromatic gems are self-colored. They absorb and reflect certain light waves because of the characteristics of their chemical structures. Idiochromatic gemstones are those who only appear in one color. Sometimes, the intensity of the color varies due to the varying amount of elements present, but still, the color remains unchanged. An example would be a chrysolite or peridot. When we look at its chemical formula, iron, the element responsible for its green color, is essentially there. If we take out the iron, then it's not going to be a peridot anymore. Idiochromatic stones are rare, and most colored gemstones are allochromatic. Allochromatic gemstones are those colored by so-called impurities or trace elements. An example of an allochromatic gemstone, as already given before, the corundum and its variants, ruby and sapphire. Corundum is a crystalline form of aluminium oxide, and it's colorless when in its purest form. It's the most stable form of aluminium oxide. All of its aluminium ions stable, and all its electrons are joined with oxygen atoms. In rubies, some aluminium ions get replaced by chromium ions. In order to maintain the charge balance of the aluminium oxide crystal, the chromium ions lose 3 electrons upon replacing aluminium ions. But because of the differences in properties between aluminium and chromium ions, the arrangement of the oxygen ions becomes distorted, and so the energy levels of the chromium ion orbitals change slightly. The differences in energy levels will lead to the absorption of the violet, yellow, and green areas of the visible light spectrum. And that's how the ruby becomes red. The more impure the ruby becomes, meaning the more chromium ions, the deeper the red color. Sapphires are any colored corundum gemstones, except red. But mainly sapphire is mostly popular for being colored deep blue, which is due to the presence of both titanium and iron impurities in their correct valence states. Without one or the other, the sapphire will reflect a different color. For other crystalline compounds like quartz or beryl, some processes may vary, but a transition metal ion, replacing some metal ions of the original structure of the crystalline, tends to slightly alter the structure of the gem and the energy differences between the trace elements, causing the impurity with the metallic element originally comprising the crystalline compound. This will result in absorption of some wavelengths in the visible light spectrum. The intensity of the hues in the gemstones varies, depending on how much trace element is present. Mechanical plant defenses. For something that cannot speak, plants sure do have a lot of enemies wanting to hunt them down. There are annoying bacteria and microorganisms that latch onto them and kill them slowly. Insects who eat leaves or suck the nutrients out of their leaves. And bigger animals that need to eat plants for nearly their entire diet. Unlike animals, plants are incapable of fleeing from their predators. So, to protect themselves from being harmed or eaten by herbivores or plant-eating organisms, plants need to have a bunch of set-up defenses, incorporated into their morphologies. These plant defenses range from the most conspicuous to the most minuscule details we don't even notice until we get a closer look. The biggest and most obvious ones, of course, are the sharp thorns, prickles and spines that explicitly warn bigger animals that plants with thorns are not very convenient to eat. These sharp things are generally called spinous structures. Spinous structures are made to defend the plant species from herbivores. And since it's already part of the plant structure, it's considered a mechanical defense as compared to those that send out chemicals to trigger some defense mechanism. Now, of course, the more exciting plant defenses to learn about are those that are more inconspicuous. There are some leaves that feel hairy and rough because they have these little things called trichromes. Trichromes come in different appearances. Some are prickly to hurt the insects that land on their leaves. Kidney bean leaves are used to trap bedbugs because its trichromes stab the legs of the bugs. Some trichromes are not prickly, but they're sticky to make sure bugs get trapped. It's what carnivorous plants, like the Venus flytraps and sundews, have to help them catch little insects. Some trichromes are also capable of releasing chemicals, like the stinging nettle exuding histamine and other toxins that cause inflammation. When humans encounter leaves with trichromes, it probably won't hurt as much, but we'll feel that the leaf's texture is rough, or it causes mild itching on our skin. Rhaphides are another stinging defensive structure in plants. But it's a more delayed mechanism because it'll be experienced by the attacker after the attack. Rhaphides are considerably more terrifying than trichromes, because these can poison herbivores. They are like tiny needles, cutting into and tearing the lining of the mouth, esophagus, and gut. What's worse, rhaphides often contain distasteful and toxic proteins. The cuts and tears caused by the rhaphides allow the toxins to enter through the wounds, and this will cause a burning sensation in the mouth and stomach. If compounds have enough toxicity, it causes more serious problems for the herbivores. Rhaphides are not produced equally in different sections of the plant. Typically, stems tend to have more rhaphides than leaves. The number of rhaphides also varies from species to species. The shape and quantity differ, but just because some plants have rhaphides doesn't mean they're poisonous. Plants like pineapple, kiwi, spinach, and rhubarb are edible. Only some parts, such as the stems or leaves, should be avoided. Another kind of mechanical defense is called thigmonesty. This time, no one has to get pricked or poisoned, for all the plant does is to make itself look unappealing and dead-looking, so plant eaters won't bother eating them. Plants like the Mimosa pudica have special mechanoreceptor cells detect touch from approaching predators. The movement is associated with changes in the turgor pressure within the cells. When an insect lands on the thigmonastic plant, say the mimosa, the mechanoreceptor cells will send a signal from the leaf down to the stem, then to the base. The signal causes a decrease in turgor pressure, losing water, thus deflating the plant cells a little bit. That's why the leaves of the mimosa shrink or close. The way plants exhibit to protect themselves are incredible and sometimes taken for granted by humans, partly because we really don't get affected. But nevertheless, a great demonstration on how different species work really hard to survive. That's all the time we have for today. But before we go, let's take a quick recap about the things we talked about in this episode. Iridescence is the phenomenon where surfaces reflect a multitude of colors at once and change color perspectives upon shifting the angle of viewing. Iridescence is caused by interference, where light gets reflected or cancelled out by the structures of the surfaces. Chemiluminescence is the phenomenon of producing light energy through chemical reactions. The byproduct of a chemiluminescent reaction, instead of heat energy, is a photon or a light particle. Only selected compounds are capable of producing chemiluminescence. Usually highly oxidized compounds when reacting with another compound. Bioluminescence is a form of chemiluminescence, only that this occurs in living things. Special compounds, collectively called as luciferin, oxidizes upon catalysis by enzymes called luciferase, producing oxyluciferin. Upon decay, oxyluciferin releases photons or light particles. Bioluminescence serves different functions for each organism, but all for survival purposes. A gemstone acquires its color through elements that are essential to its structure, or through impurities, or elements that replace some of the metallic ions in the structure that will affect which colors in the visible spectrum get absorbed or reflected. Finally, various mechanical plant defenses demonstrate how survival instincts are incorporated into plant morphologies and physiologies by growing or producing various defensive features, such as thorns, spines, and prickles, to the less obvious ones like trichromes and raphides, and less aggressive forms of defense like thigmonesty. In our body trade a stimulus in the rubber hand experiment. Autokinetic effect. Do you sometimes see lit objects moving along in the dark? It's just another case of your eyes playing tricks on you. Let's take a look at the autokinetic effect. The autokinetic effect, or autokinesis, is the visual perception where a non-moving lit object or light source appears to move when viewed in relation to a dark and featureless environment. The effect occurs because the fatigued muscles of our eyes cause some minor motion in the retina. And given that the light source or object has no defined reference points in terms of size, position, and vantage, our brains are led to perceive some movement. The effect can also be influenced by suggestion. When you view a lit object without reference point, you are more likely to be prone to suggestion with regard to movement and position. In 1799, Sir Alexander von Humboldt observed this phenomenon and thought he was witnessing moving stars. He used the term Sternschwanken to describe it, which means moving or sliding stars. It wasn't until 1857 that G. Schweitzer, a German psychologist, would discover that the autokinetic effect was a subjective perceptual phenomenon completely unrelated to motion. Here's an experiment that you can try at home. Use a small candle or a small battery-powered light. Place it at eye level and measure its distance to you. This will be your starting point. Turn off the lights and try to walk halfway from your initial starting point. Turn on the lights and mark your position with a piece of tape. Turn them off again and try to walk halfway. Try it out with different people. In 1935, a study conducted by Muzaffar Sharif showed that while people tend to overestimate the position of stationary lit objects in the dark, they still tended to agree on a somewhat similar perceived position. That is, after participants of the study were allowed to talk and discuss with each other. The study concluded that people tend to doubt their own observations and seek explanations when experiencing the autokinetic effect. Here's another example that you can try out with your friends. Go out at night and look at the stars. Focus on one star and point it out to your friends. Suggest that the star is moving and trick them into believing you with the autokinetic effect. Of course, don't be a bad friend and explain to them afterwards the star didn't move, but rather they only perceived it to move. On a more serious note, however, pilots should always be wary of the autokinetic effect, lest they start seeing moving objects in their view. To ease its effects, pilots actually do a variety of exercises to overcome the autokinetic effect. These include constantly shifting their gaze, using landmarks like towers or buildings as reference points, periodically moving their heads, eyes, and limbs, and monitoring flight equipment to resolve any conflicts in perception. Some researchers attribute a number of UFO sightings to the autokinetic effect as well. If you think there's some connection to perceiving moving lights in the sky with the autokinetic effect, then you are correct. After all, most descriptions of the phenomena of UFO sightings fit well with this effect. Consider this familiar scenario of alleged UFO sightings. There are stationary lights in the dark night sky, and then after a while of staring at these, eyes get tired and then the autokinetic effect kicks in, thus creating the effect of perceiving a moving, unidentified object in the sky. So the next time you think you see something move in the dark, don't worry, you're not going crazy. You're just experiencing the autokinetic effect in action. Moon illusion. Here's an illusion that you can observe by going outside on specific nights of the month. What is it? Well, step outside at night and take a look at the moon. On some nights, it would seem that the moon is really far up in the sky and looks very small. Yet on other nights, the moon appears to be close and very large. Records dating back as far as the 4th century BC show that people have been observing this apparent size-changing illusion. Back then, people thought this illusion could be attributed to magnification caused by the Earth's atmosphere. But you know what? The moon's size never changes. And this is where the illusion comes into play. So, let's take a look at something called the moon illusion. The moon illusion is an optical illusion where the moon appears to be larger when it's nearer the horizon. But how do we know this? Well, if you measure the moon's size using an instrument called a theodolite, it'll appear constant, even as it moves from the horizon up into the sky. In fact, if you look at photographs of the moon at different positions in the sky, its size appears constant. You can check this out for yourself using a simple technique. On nights when the moon appears bigger, take a small object like a pebble or a coin. Close one of your eyes and try to block the moon with a small object. Now, as the moon rises, do the same again with the pebble or coin. Notice that the moon still appears as the same size in reference to the small object in your hand. So, if the moon's size is constant, then why does it seem to change in size? Unfortunately, the reason behind it is still widely debated. One of the most accepted theories suggests that the moon appears bigger when viewed on the horizon, because our eyes are able to see the moon with other visual reference points, like buildings, trees, and mountains. Because of this, our brains are able to compare the size of the moon with the sizes of these reference points. But as the moon appears higher in the sky, we lose our visual references, and therefore we don't have anything to compare it with in the dark sky. Some compare this size distortion to the optical illusion called the Ebbinghaus illusion. Observe. Which of the two middle circles appear bigger? It's the one on the right, surrounded by the smaller circles. But if you take each of the center circles and place them on top of each other, they are, in fact, the same size. Our brains are so used to picturing smaller objects in the horizon as farther and smaller in appearance. And because of this, we perceive the moon to be smaller as it appears higher in the sky, and similarly larger when it is close to the horizon. Still not convinced? Well, here's another trick that you can use to measure the size of the moon. Note that you can only do this at night during the full moon. You'll need a piece of paper. It doesn't matter if it's new or recycled, as long as you can roll it along a table. You'll also need some tape. Now roll the paper into a tube and view it at one end as if you were viewing a telescope. Look at the sky and adjust the roll of the paper to fit the view of the moon. This will serve as a size reference. Now, use this paper tube to view the moon at different intervals, and you should see that the moon never changes its size. Geometric illusions. We've been featuring lots of perceptual illusions. Some simple, some complex, others naturally occurring, while others are man-made. Now, let's talk about a form of illusion that literally looks simple on paper, but upon closer inspection, reveals some mind-blowing tricks. Let's talk about geometric illusions. Basically, these are the illusions that make the viewers perceive different geometric properties than what is actually presented to them. When we talk about geometry, we usually focus on properties such as the position of points, length, orientation, and curvature. So naturally, when it comes to geometric illusions, there is usually some form of illusion that tricks the brain into perceiving these characteristics differently. Now, let's look at some simple geometric illusions. Let us start with something simple. This is called the Delbow illusion. Here are some circles. Can you tell just by looking which of the inner circles are bigger, left, or right? Well, actually, they are the same. The appearance of the outer circles affect how our brains perceive the relative size of the inner circles. Here is what's known as the herring illusion. Watch as two parallel lines are drawn on top of these other lines that converge on a vanishing point. Do the parallel lines seem curved? It does look like it, yet they are parallel and straight. Here's another one called the vertical-horizontal illusion. Can you tell which of these two lines is the longer line? It appears to be the vertical line. Yet, on closer inspection, they are indeed the same length. This other one right here is known as the Ponzo illusion, formulated by psychologist Mario Ponzo. The illustration illustrates how our minds perceive an object's size based on the background. In this example, two lines are placed over a long railroad that is orientated toward the horizon. The line nearer the converging tracks seems to be longer than the other, but if you measure them, they are, in fact, the same. Let's check out one more example. Here is the Canizza triangle. Focus. Did you see a white triangle in the middle? You can perceive it, yet it isn't really there. How about this other triangle? It seems to be rendered in a three-dimensional manner. But look closely at the edges. It simply cannot exist in a three-dimensional world. Yet, here it is, being represented in a 2D drawing. This triangle, known as the Penrose triangle, is an example of an impossible or unrealizable object. An object that can be drawn or rendered in a two-dimensional manner, but is in fact impossible to make in 3D. So why are we being tricked into seeing something else? Well, it has something to do with how our brains and our bodies work. In psychological terms, some optical illusions are caused by the effects of excessive exposure of our eyes to stimuli. Example of these are after images caused by strained eyes. Here, look at this box for a few seconds. Now we'll show you an empty background. See anything? We are able to see the after image because of the strained photoreceptors in our eyes. On the other hand, some illusions are perceived because our brains are making unconscious inferences as it tries to make sense of an illusion. An example of this is the Necker cube illusion, where the brain tries to fill the gap in perceiving the position of the black and white blocks. Do the boxes appear straight? Do the lines appear angled? Let's take a closer look. On closer inspection, the boxes and lines are actually straight. It's another case of your brain trying to cope with what you are seeing, and thus making you perceive the illusion. Stereoscopy. Have you ever tried using one of those 3D glasses where you can see pictures and videos pop up from a flat screen? Now, do you ever wonder how they make these images appear in 3 dimensions? Well, you're in luck. Today, let's take a look at the process and technique for making flat visuals appear in 3D. Stereoscopy is a term for the technique of creating the illusions of depth by means of binocular vision, or sending two slightly different images to each eye at the same time. The term comes from the Greek stereos, which means firm and solid, and scopio, meaning to look or to see. But why do we see the illusion of depth? Well, it has something to do with how we see. Our eyes each perceive a slightly different image when we look at things. Close your left eye, then close your right eye, and you'll notice a difference in what you see. When our brain receives the images that each eye sees, it resolves this by perceiving depth. So, by allowing each eye to view a slightly different image, you are tricking the brain to perceive depth. 3D viewing and stereoscopy have been around for quite some time now. And you'd be surprised at the different ways people accomplished this throughout the years. In the 1800s, stereo images, or stereograms, were actually pictures taken side by side at the same time. A makeshift viewer usually consisted of two lenses, providing a way to show each image to each eye. Take a look at some of the old stereograms. Actually, you don't need a viewer to experience the depth effect. A crude technique you can do is to squint your eyes as you observe the stereogram. Why not try it? In the 1950s, a type of stereoscope viewer known as the Viewmaster became popular as a kid's toy. It used transparent photographs arranged in a disc to show 3D images. Other ways to view stereoscopic content, like movies and videos, involved the use of specialized glasses. These needed a way to separate the video viewed by each eye. This was accomplished using different methods, like the anaglyph method and polarized method. Some 3D viewers use motorized shutters that move really fast, covering each eye's field of view in rapid succession. Then there's holography, which uses lasers to capture and recreate the light field of an image, essentially recreating the way light bounces off an object and reaches our eyes. And finally, there's wiggly stereoscopy, which is a crude technique that involves rapidly shifting the images for the left and right eye, creating a slight effect of depth. And while 3D as a novelty will find many applications for arts and entertainment, fields such as medicine, education, and space exploration are starting to find applications for stereoscopy for training simulations and actual operations. Virtual reality, or VR, is a practical application of stereoscopy. It is used in conjunction with speakers and glasses that are used to isolate the senses of its users. Astronauts in training are using VR to conduct simulated space missions right here on Earth. Pilots of sea and air vehicles are also utilizing VR to familiarize themselves on different scenarios and operating conditions. Surgeons use VR to train on virtual patients to avoid the risk of injury and allow them further study before going on actual surgical operations. So, expect 3D to be more common outside of movies in the near future. That's all the time we have for today. But before we go, let's take a quick recap about the things we talked about in this episode. The body transfer illusion is the illusion of perceiving another object to be part of your body. This is best exemplified by the rubber hand experiment, wherein a volunteer is shown a rubber hand while their real hand is obscured. Both the fake and real hand is subject to the same stimulus, and soon the volunteer will perceive the fake hand as theirs. The autokinetic effect is the effect caused by subtle movements in our eyes when looking at dark environments. This causes stationary light sources to be perceived as moving, even though they aren't. The moon illusion is the illusion where the moon appears to change sizes during different periods in time, especially when it appears closer to the horizon. In fact, the moon's size remains constant. This happens because we are able to compare the moon's size with visual references, like trees and buildings, when it appears nearer the horizon. Geometric illusions are illusions that can literally be illustrated on paper. This type of illusion makes use of geometric properties of renderings and illustrations, and makes them appear different in terms of points, length, and curvature. Stereoscopy is the technique wherein our eyes are made to perceive two images that make up one whole image. This technique enables us to perceive three dimensions from two flat image sources. Stereoscopy has many applications outside of movies and entertainment. Among them are training simulations in the form of VR for fields as varied as space exploration and surgery. In our